So it's my honor to introduce Dr. Lauren Albrecht. She's at the University of California, Irvine, and is assistant professor. She got interested in studying the kidneys when she herself was diagnosed with a kidney disorder. And so she is going to explain to us the work she's been doing at the Albrecht lab, the research her team and herself have been working on. So without further ado, Lauren.
Thank you so much for that very nice introduction and for having me. I'm really excited to be here. This is the first time I've presented to this community, so me and my team, we're really looking forward to hearing any questions you might have and just getting your feedback. So our lab studies methylation. So we're really interested in understanding lysosomes. So lysosomes, as we've heard now many times in all these wonderful talks, lysosomes are in fact the trash can of the cell. They are in all of our cells, and they're very important. And they do more than just their trash can capabilities, where they actually serve to restore nutrients. They break down proteins, so that's kind of the trash can component. And then they also dictate cell signaling. So I'm going to touch on a few of these aspects of lysosomes and how they connect with cystinosis today. Okay, so zooming in even more, I found out I love a zoom in, but it's because we actually do study the molecular mechanisms of things. So let's zoom in even closer to the lysosomal membrane. And here's a protein we've all heard a lot about as well, which is cystinosin. That's a transporter that is in the lysosomal membrane. And it is, so here we have the lysosomal side. Cystinosin transports cysteine through the lysosome out into the cytosol. And in the case of and where it is then converted into its functional form. And in the case of cystinosin, we have a dysfunctional or absent cystinosin transporter, and that leads to the accumulation of cysteine inside of the lysosome. And so we study the context of cystinosin and lysosomal disorder in two models today that I'm going to talk about, and that they are affected and very important in cystinosis, which are the kidney and the bone. So I'm going to tell you a little bit about our kidney story first, where we're focusing on the proximal tubule cells, which are very important. And so this is in collaboration with Dr. Sergio Katz, who shared his proximal tubule cells with us and was performed by a member of my team here, who's Stephen. And so in my lab, we think a lot about visualizing cells and the molecular mechanisms. And we wanted to visualize lysosomes in the context of cystinosis. And so just to orient you, these are proximal tubule cells, and they've been these are human proximal tubule cells stained with an antibody for lysosomes, LAMP1, and they are kind of distributed here around the nucleus, which is labeled in blue. And in the absence of cystinosin, so a cystinosin knockout cells, when we're looking at our lysosomes up close, we see that they are enlarged, which is consistent with previous reports. So here I just have our little cartoon of cystinosin. So we found that the lysosomes were enlarged, which is consistent with the literature up to this point. And it really got us thinking about what's kind of going on with these enlarged lysosomes. As we've also heard during this conference, there have been many reports showing that the processes in which proteins are delivered to lysosomes, known as autophagy, that these processes are misregulated in cystinosis. Yesterday we heard from Dr. Katz about chaperone-mediated autophagy, and from others about autophagy or macroautophagy, but there's actually a third autophagy process for delivering proteins into the lysosomes that had yet to be investigated in cystinosis, and that is microautophagy, or the littlest autophagy, visualized here. And that is what we study in my lab. My lab in particular studies the delivery mechanisms of protein entry through microautophagy. And this is occurring through a protein modification, which is seen here as a little pink circle. But this protein modification is a methylarginine degron, or Mr. Degron, if you will. And so this is what Mr. Degron looks like. And so when we have Mr. Degron modified proteins, this leads to a rapid delivery mechanism and entry into lysosomes where those proteins are degraded. And so we wanted to investigate whether this very important process that we've discovered is essential for mechanisms during development and can become awry in other diseases, if this mechanism could be playing a role in cystinosis. So we took our proximal tubule cells, stained them with our lysosomal marker, as mentioned before, LAMP, and also stained them with an antibody that specifically recognizes these Mr. Degron targets, that it binds to methylated proteins. And we see that, so the cells are indicated just by these lines, and then the nucleus is indicating different cells, which is in blue. It was very striking what Stephen found when he was doing this experiment. So we actually didn't know which way we thought if methylated proteins would be in the lysosomes, maybe less in cystinosis, but what we found was the complete opposite, was that there was if you compare the top with the bottom, in the absence of the cystinosin transporter, there's a massive increase of methylated proteins, and these methylated proteins are entirely sequestered inside of these lysosomes here. So this seems to be an affected mechanism in the absence of a functional cystinosin. And so then we wanted to see, okay, what if we put on the frontline treatment, cysteamine, into this context? So here we have a little schematic. So we added cysteamine to these cells. And so as we all know, cysteamine, it makes it so that in the absence of a functional cystinosin, we have cysteine can form a bridge with cysteamine, and that enables it to exit the lysosome through pick lock. So we treated our cells with cysteamine, and unfortunately, it looks as though the methylation was not rescued at all. So it remained entirely stuck inside of the lysosome in the absence of cystinosin, with or without cysteamine. And so this is really important because we're always trying to understand how we can improve and build in new directions. And so understanding the molecular mechanisms that are resistant to cysteamine or not changed could tell us more about how we can design newer drug targets. So just to summarize this, what we have here, a healthy cell, we have proteins, so this is just a lysosome inside of a cell, right? And so we have proteins, they get their Mr. Degron modification, they are delivered into the lysosomes through microautophagy, and they're broken down in a healthy cell. Whereas in a cystinotic cell, the proteins that are methylated, they go through microautophagy and they get into the lysosome just fine. But the problem is that they accumulate there. And so so we have this accumulation of this asymmetric dimethylarginine. So what I didn't mention was that when proteins that are methylated or modified by Mr. Degron, once they undergo degradation inside of the lysosome, they actually lead to the formation of this free metabolite, which is asymmetric dimethylarginine. That's because the methylation group, it's not actually removed from the residue that it's modifying, it remains bound to it, but the protein is still chopped up. But so what you get is this free ADMA molecule once the protein is degraded. Now this is really important because you're wondering, why do I care that I have a bunch of methylated proteins inside of my lysosomes, maybe? Which is what we were thinking. Like, how can we actually use this information, this really striking result, how can we use it? And so ADMA, in the context of a healthy cell, it's actually doing a lot of things. This small metabolite, it actually has a large span of binding partners. So this is a list of, these are different proteins that their activity is regulated by ADMA. So ADMA, this metabolite, it binds to these proteins to regulate a variety of different major cellular processes in redox control, glycolysis, metabolism, growth, energy storage, cellular detoxification. So this is leading us to wonder whether in the case of cystinosis, where we have this accumulation of ADMA in the lysosome, and as such, a low level of ADMA in the cytosol, could this be impacting these downstream signaling pathways? And so this is an ongoing, active area of research in the lab, and you can feel free to talk to my team. They're here today about if you have more questions about this. But as an initial target, so here I have our hypothesis. As an initial target, we went after nitric oxide synthase, as it's a very important protein for regulating vasculature and various other aspects of cell metabolism. And we took our proximal tubule cells where we have a normal high level of ADMA in the cytosol because it's being degraded normally and released normally from the lysosome. And we stained it with a fluorescent marker of nitric oxide activity. And so we see there's kind of basal levels, whereas in the cystinosin depleted cells, we now see that there's an accumulation of this DAF where there's low ADMA. We actually have high activity of nitric oxide synthase, which is expected because ADMA actually inhibits NOS, nitric oxide synthase. So this was an exciting result for us, and so we're going to continue along this molecule in addition to the others that I mentioned in the previous slide. So as a summary for the first part of my talk, we have Mr. Degron is a new autophagic pathway that's implicated in cystinosis, that it is misregulated in a way that is resistant to cysteamine treatment, and that perhaps this could help us to understand new molecular targets that are regulated by this important metabolite, ADMA. Okay, and for the second part of my talk, I'm going to get into bones a little bit. Okay, and so bones and kidneys are talking to each other, so they're really kind of important together. But this is a cartoon depiction of a bone. We have 3 different bone cell types. We have our osteoblasts, our osteoclasts, and our osteocytes. It's easy to remember what these 3 bone cell types do because osteoblasts build bone, for B. Osteoclasts, they chew the bone, so they break it down all the time. And then osteocytes are just special. So I'm going to talk a lot about them. But basically, our bones are constantly remodeling and turning over all the time. And it's very important that we have a regulated activity of our osteoblasts that are forming new bone and osteoclasts that are resorbing bone or breaking it down. And that's how we regulate the level of calcium in our blood. And in the context of cystinosis, we have a misregulation of these osteoblasts and osteoclast activity. And this is a patient staining of the inside of a bone, some histology. There's increased osteoid, there's altered bone turnover, there's mineralization defects, and all of the symptoms that I'm sure we've heard a lot about. And so while the literature has focused primarily on understanding the role of osteoblasts and osteoclasts in cystinosis, which is still not very well understood, we're still working on it, the osteocyte has received much less attention, and that's because of its difficult, challenging location being inside of the bone matrix. So osteocytes, this is inside of a calcified bone matrix. We have these individual osteocyte cells. They're very special. They have all of these dendrite projections all over the place, and it's because each one of these cells that wants to be talking to each other, they can't, like in other tissues, because they're inside of a calcified bone matrix, so they form these projections. So these are really important for signaling inside of the bone. And so osteocytes actually secrete important proteins that go through these structures, these canaliculi, and they reach the bone surface to regulate both the activity of osteoblasts and osteoclasts. So they're actually sort of a master regulator of bone formation, turnover, and resorption. And so we wanted to look at these osteocytes, and we wanted to do that in the context of cystinosis. And so here we have, I'm going to be showing you first bone that we stained from a healthy patient and comparing it to the bone of a cystinotic patient. So just to get you kind of oriented, we wanted to look at lysosomes in particular because we're obsessed with them. And so in the healthy bone, we have our LAMP1 staining. And so this line demarks, this is bone marrow over here, and this is the calcified bone matrix, so inside of the bone. And these are little osteocytes, and this image corresponds to the image below that's simply marking the nucleus, so just kind of where cells are. So if we kind of do a zoom in here, we have our osteocytes inside of the bone matrix, and you can actually see these projections coming off of them. So they're very high in lysosomes. So now we're comparing this to the bone matrix from a cystinotic patient, again marking here's the bone matrix, and here's where we should be seeing a lot of osteocytes. And in fact, we do see the nucleus, so the cells are present actually. But what you can see is it's quite a striking difference. Even zooming in close on our cells, we see that in the case of cystinosis, these cells have very low levels of lysosomes. And this was a really striking result for us. And so we wanted to see whether this was happening in multiple patients. And so there are very limited bone tissues from cystinosis patients. But what we found was that this was very consistent across 3 patients where we're quantifying as many cells as we possibly can, that they had a dramatically reduced level of lysosomes. And we did it with multiple markers of the lysosome, both LAMP1 or LAMP2A. These are 2 different proteins, both that mark lysosomes. They were consistently down in cystinosis. So this was really interesting. To further confirm this, we in a collaboration with the Sharkey lab, we obtained the cystinosis knockout mice, and we see that compared to the wild-type mice that have nice lysosomes in their osteocytes, that the cystinosin knockout mice had a dramatically decreased number of lysosomes in these bone cells, in these osteocytes. And we also confirmed this at the transcript level by doing qPCR to show that the levels were lower. So this made us wonder about what the lysosome is actually doing in an osteocyte, and how could this be playing a role in the bone defects of cystinosis. So I mentioned that osteocytes are kind of the global regulators of osteoblasts and osteoclast activity. And how do they do this? Well, thank you. Perfect. They secrete proteins, and these secreted proteins, they actually can regulate the osteoblast or osteoclast activity. And so one of these secreted proteins is sclerostin, and sclerostin inhibits bone formation. And so that's important. It's secreted, bone formation is inhibited, and And that's great. Okay, so how do we want to turn it, if we're secreting sclerostin now, if we want to turn bone formation on, we need to turn sclerostin off. How do we do that? The cell actually degrades sclerostin. And so zooming in on our lysosome that we know and love, sclerostin, the secreted factor, is actually targeted into the lysosome and broken down. So this is the mechanism of turning sclerostin off. So that made us wonder, in the case of cystinosis, where we don't have functional lysosomes in our osteocytes, could this lead to an aberrant upregulation of these secreted factors? We can't turn them off. They're just secreting all the time. And could this be a way in which there's just too much bone turnover? And so, this is an ongoing investigation, but when we looked at our control versus cystinosis, it was evident that there is an increase in sclerostin in these osteocytes in the case of the cystinosis model and not in the control, suggesting that perhaps this is the case. So, to summarize part 2, osteocytes, maybe they are really important for secreting factors. These secreted factors, they offer both new therapeutic targets that are currently being used for treating osteoporosis in different contexts, and also biomarkers. So it's really important, these secreted factors from osteocytes, as new angles to investigate. And we have a little bit more understanding of these osteocyte cells and what lysosomes are kind of doing and functioning in them in the case of cystinosis. And so now I'd just like to acknowledge the collaborations that I have with amazing scientists in this room, from the Shirky lab, from Sergio Katz's lab at UCLA, and the team that is with me doing this research. And this is a QR code for our lab website if you want our contact or to learn more about the research that we do. And I want to thank you all for your attention and for having me here. Thank you.
Thank you so much. That was really interesting and new information for us. So I think we have time for maybe 1 or 2 questions, if anyone in the audience has any.
Bear with me because I'm an accountant, not a scientist. But in terms of the lysosomes in the cystinotic bones that you looked at, were they adult patients?
Yes.
They were.
Okay, the question is, what is the age of the patients that we were looking at their bones? They were teenagers.
Okay. So would you anticipate more lysosomes in younger bones, and it's a progressive?
That is such a very, very good question. That is what I would anticipate. Obviously, things are crazy, you know, when you're studying cystinosis, there's all these unexpected things, but that is the hypothesis that we would expect right now, yes.
Great, thank you.
Thanks. Yeah.
Anybody else?
So I always sort of visualize the cells of patients with cystinosis to be chock full of these poorly functioning lysosomes that are stuffed with cystine. Why aren't you finding any lysosomes in the damn bone cells? Like, that seems so weird.
I completely agree. I think that it's also very weird. The question is, the case is that lysosomes are engorged and large, and so what are you telling me that in these bone cells there are no lysosomes? So we're wondering this, you know, what's happening here? So if we're comparing different bone cell types, so these are the mature osteocytes, we suspect it could be something having to do with the half-life of these particular bone cell types, in that, you know, they're one of the osteocytes live for decades, if it's something to do with that, because obviously it's not the case at all in most other tissues. The lysosomes are completely there. So the result to us is extremely interesting and unexpected. Yeah, so I think but they are these mature osteocytes.
So, it's a follow-up question on, you show that at the RNA level you have a decrease in LAMP1 and 2A. So you think it may be a defect in osteocytes at the transcriptional level on the lysosomal biogenesis signaling pathways through TFPB or other?
Yeah, it could be. Yeah, the fact that it's also the levels of LAMP are decreased at the transcriptional level and the protein level do definitely tell us new things about the mechanism. So it could possibly be through that. That's true. Yeah. Thank you.
All right, thank you so much.